about some new work that in particular was done here in Abu Dhabi. And so I will get to that in like halfway through the talk. I'm going to talk about regeneration, and this is my favorite topic. And here I've shown a couple examples where here's a fallen leaf that started to regenerate a root. Here's a stump that started to regenerate a shoot. And you instinctively know, because you've seen all these types of things, that plants are really, really good at regeneration. And the main question in the lab is, how do they do this? And so we use a system, or a few systems now, and I'll show you those. But first, I just want to say, since we were talking about, and we do get a lot of our funding from climate change initiatives, it's not just a really interesting question and a really interesting phenomenon, but it's really, really important for biotechnology, right? Because it's through the regeneration of plants that we get all kinds of genetic approaches to creating new kinds of crops. And this is one of our big hopes for dealing with climate change and making plants more resistant to drought and heat. And so it's quite relevant. And one of the problems is that the plants that we rely most on for food are quite recalcitrant to regeneration. They can be done in certain ways, but it's very cumbersome, and it takes a lot of time, and it can't be done in high throughput. So any means that we can use to accelerate the regeneration of these crops in particular will be very, very important to us in the near future. Okay. So our system is the Arabidopsis root, and here it is. And if you pull up a root, all you really have to know is that this structure that I'm showing you here, and I'll go into it in some detail in a minute, is the conserved structure. Basically, all the plants, all the land plants that you see have this kind of root, almost all the land plants. And here's things you learned in high school, right? The xylem, the phloem, the pericycle, the endodermis. I won't talk much about that, but that's just to say there are these concentric layers that all serve a purpose of this kind of drilling machine, which is gathering water and nutrients and bringing it to the top. But that's all because there's a stem cell niche, just like you would consider in humans or anywhere else, where all the cells are born. And so there's this region here, which is called the quiescence center, and it's surrounded by stem cells. And so those cells divide, and they gotta get pushed up here. And of course, plant cells, they don't move and migrate around, so all the development here is happening largely through cell-cell communication. But very important, we're gonna track this region wherever you see this marker, which I'm going to show right here. This is WOX5, for example, nice marker that marks the very, very center of that stem cell. And as you could tell by the name, these cells don't divide much, and so they stay put, and they, in theory, support the cells around them as stem cells. Okay, so the entire growth center is here. These cells down here are also born through the stem cells, and they're sloughed off. The plant loses these as it moves through the soil. So our system is this, where you cut off that whole growth center, right? So the stem cell is gone. Here you see the markers of WOX5. And then in a day, it reappears, right? So this is what makes plants so amazing. It's just like cutting off your head and your eyeballs kind of reappearing in your chest. But you can notice it's in the wrong place. And then by day 2 here, this, if you're a developmental biologist, right, is a really, really important point, because here now, even though there's a lot of misexpression, here now that the quiescent center has found its place, its coordinates from the bottom, right, and from the sides, it knows exactly where it wants to be. And then within another day, it is kind of very much refined. The pattern's still a little messy, but another day, and you could not tell that this root was ever damaged, right? So this is the fascinating question that we work on. We have a lot of questions. Where does the patterning information come from? How does the plant know it's injured? And we really look at cells and ask, how do cells go from one state to another, and what makes a cell competent to regenerate? And so I today, I'll address part of this patterning question. Okay, so I'm just going to give a little background for a lot of work we've done over the past 10, 15 years. And that is to say that what regeneration looks like in many ways is very similar to what embryogenesis looks like. And in particular, so here's the one-cell stage, and you can see it moving on. This is appropriately called the heart stage. But right here is a really important event that happens, and this cell called the hypophysis is going to come into existence. And then a little later, it's going to divide into this lens-shaped cell. This lens-shaped cell is where the QC is going to come from and WOX5. And in fact, all the cells of the root that are unique to the root are actually going to come from that hypophysis. So that division is very important. It marks the division that's going to give rise to the QC versus the cap. And we're going to follow that division a little bit, and that story will become relevant in a minute. Okay, so what we see in regeneration when we cut, and here's a marker for that cap area and a marker for that QC, and when we cut, and this is only about 12 hours, the plant is already starting to rearrange things, is that we see the WOX5 and this WIP4 marker that are the tip marker here actually sort of co-expressing in the same place. So while they're really in different places before you cut, now once you cut, they sort of converge. And what this interestingly resembles is the hypophysis, right? And so here are all the hypophysis markers. Now, it's not like the plant has one set of genes for making an embryo and another set of genes for making the mature root. And of course, the mature root continues to exist and divide and make more roots. But it's the arrangement of markers here. These markers are all expressed in the adult, but they're not expressed together. But here, during regeneration, they're expressed together. So this was our first clue that the regeneration process is actually really resembling, it's recapitulating part of embryonic development. Okay, all right, good. So that's where we are so far. We have embryogenesis, we have the splitting of the cell, and so now it's time to talk about the thing that comes up in every plant talk, which is auxin. It's like fairy dust. It controls every part of plant development. But here's a classic experiment from the 1950s that's really sort of set the stage for a lot of research in hormone biology and in development in plants. And here you see this was a really important step because in that in vitro regeneration process, this figured out how to actually make roots and shoots, and from there you're off and you can regenerate anything. How am I doing on time? 15? 16. Okay, that's very hard to see. Okay, so if you have high auxin concentration and low cytokinin, these are two hormones that are sort of always together and they're always antagonizing each other, but they always need each other around. They're like brothers. And so high auxin, low cytokinin, you get roots. High cytokinin, low auxin, you get shoots. Did I say that right? Roots, shoots. Okay. So, but it's not only ratios here. This was a discovery not by us, but by others that showed in embryogenesis, you first have co-expression of both the auxin and cytokinin together. Then they kind of separate. Here's a cytokinin marker. That lens-shaped upper cell takes the cytokinin. And then later, at some point, it's going to express the auxin. So there's kind of a sequence to this. So timing matters in the birth of the stem cell niche, where you get an auxin cytokinin, then you get a cytokinin environment, and then you get an auxin environment. And similar things seem to be happening in regeneration. And here I'm just showing, sorry, this is from our single cell experiment. I won't go into a lot of detail. But it's really basically showing that first stage where auxin and cytokinin are expressed together. We think then here are the markers, a cytokinin marker. an auxin marker, these are going to be co-expressed. Then the cytokinin marker is going to be only in the auxin environment, and then it's going to be auxin that sort of takes over and keeps the QC going. All right, so to sum that up, and now to get to the new parts in the next few minutes, the colors have changed. Here, right, just like embryogenesis and in regeneration, a mixture of auxin cytokinin, then you get the QC born in the cytokinin environment, and then as it matures, it's switching over to a new hormonal environment. And this makes sense. Development is all about timing and what you can do. So can we use this information to try to figure out better ways to regenerate plants in vitro or wherever we want? And this is where the project by my postdoc here, Alya Hessen, comes in, and she's assisted by our technician Atik here. And the question is there a Morse code to auxin-cytokinin interactions? And so Alya's question is, does it mean something for the plant cell to be in both an auxin, so I'm using, this is Morse code, by the way. So I'm using auxin as the dash and cytokinin here as the dots. So is there a difference between simultaneous treatment or an environment where you have both cytokinin and auxin, and a sequential environment? How far back did I go? A sequential environment where you have cytokinin and then auxin. And so for this, Alya's using cell culture, right? You can take a plant, any root, you can treat it with enzymes that'll break apart the cell walls from fungi, and then you can have naked cells, which we call protoplasts, in culture. You can have cultures, you could have immortal cultures in plants like you do in animals, but we're using primary cells from the plant. And so Alya's done a series of experiments here where here the cell's just treated with auxin. Here we're measuring the auxin response just by that DR5, this reporter for auxin. And in every series here, which is a different color, you're going to just get increasing cytokinin concentration. And so just like we have seen before, as you increase the cytokinin, here we have auxin, and then we're adding cytokinin. Here we have cytokinin, and then we're adding auxin. But whenever they're expressed or whenever they're treated simultaneously, the cytokinin is inhibiting the auxin response. Just like we know in regular development and in most scenarios, and just like we think happens in this ratio between auxin and cytokinin. Then Alya tested a different situation where instead of simultaneous treatment, there was a pre-treatment with cytokinin and then auxin. And so here you're just looking at the same result you saw in the previous, where it's auxin and cytokinin together in various combinations or sequences. But now here we're going to look at, here we're going to look at the cytokinin first, then auxin. And surprisingly, this actually enhances the auxin response. So it seems very possible that this series that I showed you before of a co-environment, a mixed environment of auxin, cytokinin, then cytokinin only, and then auxin, is actually the role of the cytokinin here might be to prime this auxin response and to make it nice and robust to initiate this stem cell niche. Okay, so then we start to ask, can we create environments in vivo that initiate the stem cells? And here now I'm going to talk about some new work that Alya has done in collaboration with an engineering group here. Okay, sorry, I skipped a thought here. And what we wanted to do was create sort of mini environments. And so this is going to be work in progress. I'm not going to come to any great conclusion, but I'm going to show you sort of the technology that we've come up with. Okay, so another consideration, of course, if you are a developmental biologist, is mechanics matter, right? The tension that cells are under. And here's a nice study from just a couple of years ago that I think really gave some nice definitive proof that when you regenerate the shoot, you loosen cell walls, so you create sort of this ring around this nascent area where the stem cell niche is going to form. And it creates like a a stiff band that the cells that are going to regenerate into the stem cell niche kind of push against. And it's this tension, which they call mechanical conflict, that gives rise to this new stem cell niche, meaning you need, and they also showed that you need this tension, so if you loosen these cell walls, you won't get that to occur. So we need an environment where we can both change the chemical composition and actually restrain or have some mechanical restraints. Okay, so here's where we turn to microfluidics. And this is our collaboration right here at NYU in the engineering department between me and Raphael Song's lab, but also the main, of course, drivers are Alya and Aji Orozalev, and we're still learning how to pronounce his name, so we just call him Aji. Okay, so what we're asking Aji to do is something most plumbers would not want to happen, and I guess engineers maybe, or fluidic engineers. It's a difficult problem because we want to make a clog, right? Just like you make a clog in your drain. But we also need flow because these cells are not going to survive. It's going to get to a hypoxic environment, and they won't survive if there's no flow. So it's quite a challenge for engineers. So now I'm just going to run through how, for example, a fluidics engineer solves this problem, because it's kind of interesting and fun to look at. Okay, so here was design one. We can call it the drain pipe. Straightforward. The cells were supposed to come down here. There's a flow. They were supposed to get trapped here, but they decided to just flow out this other way, and that didn't work. We couldn't capture any cells. Okay, there's the movie. Here's drain pipe 2, where there is, of course, this is a fatter channel, and these are very thin channels, and Aji here is trying to make more flux so that we can trap the cells and not get clogged up, but this didn't work because they sort of aggregate over the vents and then they block the flow and nothing happens. Okay, version 3, which they all get funny names because of the way they look. This Apollo 11 for obvious reasons. And here Aji is really increasing the flux to really try to get the flow to come out as well as trapping cells. But we trap the cells, we clog the vents, and the cells accumulate, the pressure builds, they get squashed, they get hypoxic, so that didn't work. Okay, so there's a tough engineering problem. Okay, here's the satellite, or maybe the Martian, I don't know. And Aji had the idea that if we have the media flow in from one channel and the cells another channel, and now we have this cup-shaped catchment, we could have good flow here. And now here's a really important development in the process where we have just several micron little channels on the side, sort of vents that'll allow some flux of media in and out. Nonetheless, despite that advance, it was hard to trap the cells. They're still suffering from hypoxia. So this was an advance, but didn't work. Okay, and how am I doing on time? 6, perfect. Okay, and now the cutting board. So Aji now, of course, like you have one try and you have to load the cells, so why not have a lot of different cups here? And so here we start to load. Oh, I'll show you just a little image of the cells whizzing by. Unfortunately here, there's a lot air bubbles that build up in here and they go everywhere but inside the cup. So we had to degas, we had to treat the microfluidic device. Here's the next version, design 6, where Aji has here developed the fluted cup, which can catch the cells. And this was starting to work better, but not quite efficient in capturing cells. And now here's sort of the final version where he's carefully measuring the cells. We're getting cells to flow down in through here. They're sort of squeezing, but then when they squeeze between, they get caught in another cup. And now this seems to work. And so the cells get trapped in here. They are viable for a long time. Here's 96 hours where they stay. They start to get big, but now here we've got them trapped in this environment. Here you can see they kind of stack up. Here's viability stain, FDA. There's fewer cells in here because we have to get up and move it and take it to a new microscope. We're going to solve that problem soon. And so you can see here viable for now 6 days. So we got a long-term solution. And we can get, here's calcifluor staining. You can start to see cell wall formation. Of course, this is really, really important because plant cells will not divide unless they can form a cell wall. That's the first thing they do between the two daughter cells. So really important. Here's maize in the system. Sorry, I forgot we gave this a formal name, calling it the crib. And so it stands for a very cute acronym. And so we can pack the maize cells in, and here you see the viability stain, and they're still alive. Okay, so this is the end of the story. Sorry, I don't have any real experiments yet, but we finally think in this walk we have this nice little system where we can replicate the mechanics, right? They're kind of being squeezed here. There are some physicists who worked on the shape of the root, and there's a compressive forces right at the QC that seems to really create a lot of compressive tension there. So we think we can replicate that here. We can get the flux, we can keep them alive, and now with the fluidic system, we can change the environment sequentially, right? Where we add both auxin, cytokinin, just auxin, just cytokinin, then auxin. And so we have a lot of hope on this. All right, so conclusions, regeneration looks like embryogenesis. We can recapitulate the complex hormonal environments. It's all about change. It's all about switching over time. So now we have the device to do that, and I hope, the next time I talk, I can tell you some cool things that are happening there. Okay, so first, I want to thank everybody. I think I've mentioned everybody here. Bruno Guillotin, he's a New York guy who comes to Abu Dhabi a lot. It's been a great contribution to this and other projects. And I do want to also thank Ines, who's been mentioned a couple of times here. She runs the show and does all kinds of cool things, like put this on the web and in social media with us on the wave here, me kind of cresting, which is appropriate. And our supporters here. And my final thing, which I've used up of my few minutes here, is I do want to give a plug. Alia has created this UAE Plant and Environment Society, where we are, if you're a plant scientist out there and you're in the Gulf area, not just UAE, we hope you'll join with monthly meetings that we're going to plan out and try to get a group together that's thinking about drought, heat, and other stresses on plants in this region. Okay. And then with that, I will take any questions you have in the 2 minutes I have remaining. And so thank you.